Okay, so here's a caricature of how drug R&D works. It's not my caricature, it's the industry's caricature. If you ask the UK Trade Association or the US Trade Association, or if you read textbooks, or if you do master's courses on biomedical innovation, this is the chart you get shown. And I think there is an implicit model of how therapeutic innovation works, which is not always made explicit in this process. So there are a bunch of grey arrows, and those grey arrows represent various individual scientific activities. I won't go into them in any detail, but towards the left-hand end, we've got bits that are around identifying bits of biological machinery that you think have gone wrong. Towards the middle, you've got processes which are around finding chemicals that you think intervene on those bits of biological machinery. And then at the right-hand end, you've got testing in people to see if it works. And I think there is a logic implicit in this process, or explicit in this process, that it's something like this. Diseases are caused by bits of biological machinery going specifically wrong. That causes some nasty symptom, which here we call Y. You can do something to that molecular component by sticking a drug on it. That will make the symptoms better. There's still a lot of diseases and a lot of symptoms out there. So therefore, if we do all of those things in the grey arrows much better and much quicker, that's going to be a really good way of discovering lots of cures for lots of diseases at low cost. So we're going to build an academic establishment in the biomedical industry around doing those things, because that will deliver us lots of great new cures at low cost. Okay, that's the logic. And in terms of input efficiencies, if I think about how cheap and easy the things in the grey arrows have become to do, this has been a triumph, right? So one of the grey arrows was target identification. Genomics is important for that. Well, it's over 10 billion times cheaper to sequence DNA now than it was in the 1970s. You could say very similar things for looking at protein structures to design drugs to stick against them, to make screening libraries. All of the things that everyone who does drug R&D say are important for doing drug R&D have got hundreds, thousands, millions, or billions of times cheaper. Fantastic. But if we look at the quality of published academic research, people working in the drug industry now, when they try to replicate academic work that was produced using these incredibly efficient, cheap, high-quality methods, they find that most of it turns out to be false or irreproducible. And if anyone funding rare diseases wants a good education on this, there are some wonderful papers on terrible mouse models of motor neuron disease, which suggested every drug that's gone into human trials in motor neuron disease, it's not that it worked in mice, then it didn't work in people, it didn't even work in mice, right? So for some reason, lots of academic stuff just seems to be wrong. And although the inputs into drug R&D have got individually hundreds, thousands, or billions of times cheaper, if you look at the amount the drug industry spends per drug discovered, it's gone up roughly 100-fold in inflation-adjusted terms since 1950. And you can see there are some things that influence this trend. Interestingly, the great biomedical revolutions and technologies haven't influenced this trend very much. So, for example, the first wave of biotech drugs didn't seem to make much of a difference to the trend. Lots of genomic technologies didn't seem, although perhaps more recently they have made a change to the trend. Regulation clearly makes a difference. You see the trend dipping in the 1960s following regulations around thalidomide that made you test to see if drugs actually worked before you gave them to people. Before that, you just had to essentially show drugs weren't poisonous, you didn't have to show they worked. There was an uptick in the 1990s when some FDA regulations were changed. And there has been a little uptick recently, which I think is real, which I think is to do with the fact that genetic slicing and dicing has meant that some of the discovery tools are working a bit better, particularly for rare diseases. But nonetheless, there's an uptick recently, but boy oh boy, it's a small uptick on the end of a 100-fold decline against a logarithmic vertical axis, right? That's units of multiples of 10. It's not a linear axis. It's got roughly 100 times more expensive. So how could this possibly happen? How could you have a drug discovery process where all of the things that everyone who does it thinks are important have got hundreds, thousands, millions, or billions of times cheaper, but the process has got 100 times more expensive? Right, there's two broad classes of explanation. One is on the right-hand side. This is, I'd say, it's a bit analogous to coal mining in South Wales. Doesn't matter how good your South Wales coal mining technology is, there ain't no more coal in the ground. And that's represented on the right-hand chart, which is a guy with a Tasmanian wolf. So imagine that drug discovery is a bit like Tasmanian wolf hunting, right? It doesn't matter how good your technology for hunting Tasmanian wolves is now, because there aren't any left. The last one died in 1934. The other set of explanations is on the left-hand side, and there's an alchemist trying to turn lead into gold. Right? Now, in this particular case, it doesn't matter how much, if he does high throughput turning lead into gold, it ain't going to work because you can't use chemistry to turn lead into gold, so actually he's adopted the wrong set of activities. So there's two broad classes of explanation. Right? One is, we've run out of things to discover, so it's 100 times more expensive despite the fact the inputs are much better. Or the other is, actually we've abandoned a whole bunch of things that used to work, and we've replaced them with things that are superficially efficient, but which don't work anymore. And the problem for most people working in drug R&D is the more objectionable you find either one of those arguments, the more you must agree with the other one. Right? So if you think one is preposterous, you are forced to agree with the other. So, why do people still believe that this is a really good way of discovering drugs? I think here is part of the answer. Every drug that is launched with a story of molecular action. Right? Drug X binds receptor Y, therefore people think if we knew enough about receptor Y and drug X, we could have predicted it. And you build this process, but what people are doing, they're confusing survivor bias with prospective explanation. And it's exactly the same intellectual error that in the US people who teach creation science make. They look at the human eye and they say, this looks so wonderfully designed, it can't simply be the product of selection acting on variation. Right? So my argument is, not all. I know some drug designers, they're very intelligent, they do great design. I know 3 or 4 people who've discovered drugs. But I think we survivor bias and the explanations that exist of drugs, I mean, we overestimate the prospective, predictive power of basic science. We don't hear, well, roughly 9 out of 10 drugs entering clinical trials fail. Every single one of those drugs will have had a really plausible explanation of exactly how it worked and why it's going to work before we can spend millions of dollars on it putting in clinical trials, yet 9 out of 10 of them fail. We don't hear the stories of the 9 that fail. Furthermore, the drugs that work, if they end up working for reasons that aren't the reasons they were originally put into trials, people simply rewrite history. And I'll show you a couple of examples of that later on. So, prospective prediction, I would argue, doesn't work as well as people think. The way the drug industry deploys its money looks much more Darwinian. Right? So what we have here is attrition. From the origin of species, Darwin said, as more individuals are produced and can possibly survive, there must be a struggle for existence. Boy, oh boy, in drug R&D, there's a struggle for existence. Right? On the left, we've got how many projects per approved drug. We saw something like this earlier today. We've got roughly 25 target-to-hit projects for each approved drug. We've got attrition at every stage. On the right-hand side, this is not a representative example, I predict a particular extreme example. We have a drug where if you go onto clinicaltrials.gov, you can see that there were 1,600 and so trials of bevacizumab or Avastin for 7 FDA-approved indications. Now, not all of those trials were funded by Roche Genentech. A number of them were just using Avastin as standard of care against which to compare other things. But it looks to me like Roche Genentech put money into roughly 350 of them. 350 trials for 7 approved indications. This does not look like precise, bottom-up, we know what this drug is going to do when we put it into people type design. As an analyst, when I worked in the city, it was very clear to me that I was not much more accurate in my forecasts than a monkey throwing darts at the Financial Times. And that is represented in this graph, which shows the ratio of sales that drugs actually achieve once they're launched to the sales that consensus, i.e. all the analysts, if you aggregate their numbers, would have said before the drug was launched. You see, we weren't particularly accurate. I was horrified to find out several years later that we were more accurate than the drug companies. Right? So we were monkeys throwing darts at the Financial Times. The drug companies were monkeys throwing darts at something much bigger than the Financial Times. Well, exactly what it was, I don't know. So this is interesting, but I'm not simply going to argue that it's all serendipity. What I think this says is that what people use drugs for when they're released into the real world are not necessarily what drug companies or the discoverers of those drugs thought those drugs would be used for. Right? It's not simply serendipity. users create use for new drugs. And in fact, there's some direct evidence of that. There's, I think, a very important and very neglected study by DeMonico and some innovation economists in the US, where they looked at all of the drugs that were approved by the FDA in 1998, and they followed them. I can't remember whether it was for a 5-year, 7-year, or 10-year period, but they followed all of those drugs, and they documented all of the off-label indications that those drugs accumulated over the following period. And of the 29 drugs that were approved in 1998, 22 of them within that period had found new uses. Sorry, 85 of them had found new uses. No, sorry. Most of the drugs found new uses, and the majority of uses were field-based discoveries by physicians. They weren't by people, so the green bar is doctors, effectively, or patients. The purple bar with 59, that is academics or drug industry. So most new uses of drugs are discovered by observant physicians and patients, or at least for this particular cohort, and not by academic laboratories or by the drug industry. And of the new uses, again, serendipity, like just, oh my God, we just saw something we never expected, there was a bit of that, but a lot of it was exactly as we saw with the insulin. It was physicians understanding the mechanisms of the drug and understanding the pathophysiology of a disease in the way that the inventors of the drug would not have done. I don't believe anyone working on mTOR inhibitors 20 years ago knew that they would have some of the uses that we now know they have. And it also struck me, I changed the title of this slide earlier today when I was listening to some of the talks. Actually, drug repurposing doesn't just happen for rare diseases, it happens for incredibly common diseases. The world's most commercially successful class of molecules are the anti-TNF drugs, Enbrel, Humira, Remicade. Those are an example of repurposing. The anti-TNFs, the world's most lucrative drug class, were originally developed for septic shock. They failed in phase 2 trials in septic shock before being repurposed in rheumatoid arthritis, Crohn's disease, psoriasis, a whole bunch of other more common conditions. Sildenafil, Viagra, we heard a bit about Viagra this morning. In fact, the Viagra repurposing story is even more complex than the one we heard this morning. The standard story for the repurposing of Viagra is it was originally tested in phase 1 trials of cardiovascular medicine, and some of the people working on the trials noticed a bizarre and potentially commercially important side effect, hence the Viagra story. In fact, another company, SmithKline Beecham, were working on a compound just like Viagra 7 or 8 years earlier. They knew exactly what it did, yet they killed the project because they thought there was no possible commercial use for that drug, and also they were worried about the reputational risks to the company if they were to be associated with lifestyle drugs. I'll point to a couple of other examples in this list. Amongst the anaesthetics, propofol, not necessarily a repurposing drug, but a really, really interesting example of user-led innovation. I should have explained this slide a bit better, but I'll explain it now. This is from a wonderful paper by Avon and Kesselheim and Avon, where they interviewed physicians to say what are the most important drugs for the last 25 years. So all of these drugs are really, really important drugs. Propofol, the most important anaesthetic of the last 25 years. The reason propofol is important is, I mean, in a sense, you think anaesthetics, how different can anaesthetics be, right? You go to sleep and you wake up again and you didn't feel anything in between. I mean, that's really all an anaesthetic has to do. Propofol, patients woke up very quickly feeling pretty good. Propofol was invented around the time that something called the endotracheal mask was invented, which was a way of intubating patients and maintaining their airway open in a rather easy and quick way, and also simultaneous with certain experiments in less invasive surgery. So propofol was used by innovative surgeons who were essentially creating day case surgery. Again, people developing the anaesthetic wouldn't have known that it would allow a revolution in the way surgery happened, because that innovation happened from the users who understood exactly how you might use an anaesthetic and how you might use it differently. There are other examples I could talk about here, and I'm going to say, but I won't. I'll give you one other example. Cardiology, lovastatin, the first of the statins. That was in clinical trials. The project was very nearly killed by Merck because a competitor product from another drug company had toxicity problems and was pulled from clinical trials. Merck cancelled all their clinical trials. Some physicians persuaded them to carry on doing trials in a very small, high-need subset of patients, and that proved the concept of the drug, and then the drug was revived and basically became the first of the statins. So user-led innovation, repurposing, is not just important for rare diseases, it's incredibly important for some of the most commercially important drugs we've ever seen. So, to summarise the problem as I see it, I think there is a sort of story told about how drug discovery works that is much less true than is commonly believed. And it's the story in the grey arrows. There is this sort of basic science push. We do lots of molecular rummaging. We predict what's going to happen. But actually, this has proved to be, I would argue, insufficiently predictive of clinical utility in man. So we've got this insufficiently predictive basic science, which then feeds into very expensive clinical trials, which are a bottleneck, again, which I would argue in many cases, maybe not so much in rare diseases, but in common diseases, are something of a game with limited real-world relevance. I came across a very interesting paper recently on depression, which argued that 80% of patients who are treated with antidepressants in the community would be ineligible for a typical antidepressant trial, right? So clinical trials often don't tell us enough about real-world patients. We then imagine that the evidence from those clinical trials forms the basis for rational health technology assessments, right? This pushes up cost and limits the number of drugs for sale. And I would argue a huge amount of innovation happens after drugs are used in the real world. So what we've done is we've got a non-predictive scientific process, an expensive bottleneck, and we put relatively little chemical diversity into the real world at very high cost. So how could it be done better? Well, Mao Zedong is rarely thought of as a good advisor on anything these days, let alone drug discovery. But he said, letting 100 flowers blossom and 100 schools of thought contend is the policy promoting progress. Now, I don't for one moment think this is going to happen. Right? I think the regulation is such, the dominance of evidence-based medicine is such, I don't necessarily expect any of this to happen. But if I was starting with a blank piece of paper, this is what I'd do. I would incentivize user-led innovation. At the moment, the biggest incentives for drug R&D are around composition of matter patents. Orphan exclusivity is good, but composition of matter patents are where the action really is. Right? Doctors are not really incentivized to discover stuff. I think that's a problem. People aren't really incentivized to find new uses for old drugs. I mean, again, orphan drug legislation helps a bit, but I think the incentives are woefully lacking. The incentives are all for doing the stuff that doesn't work very well. There's not enough incentives for doing the stuff which history suggests work quite well. I think, actually, provided drugs are safe, we need to be more liberal about what gets used, but we need much better processes for tracking what happens in the real world, so that if things are dangerous or don't work, we can get rid of them. Again, I think at the moment, a lot of this may be less true in rare diseases, but in common diseases, I think phase 3 trials are often a bit of a game. And actually they don't provide as much useful information as we want. There is a nasty, messy mixture of marketing, regulation and science, and they don't necessarily serve health systems very well. I also think there's very good historical evidence that if you reduce cycle times, progress is much quicker. Somehow, you've got to capture real-world data, feed it back into drug discovery more quickly. I think I'll leave it there and leave a bit of time for some questions.